Anybody who comes in after at this point, you guys know, automatically know it's not important. So it's my pleasure to, as we enter the summer, hope everybody had a nice graduation this weekend, bring in our second candidate for the Chan Chair and Endowed Professorship in the Department of Biology. And our candidate today is Paul Wolf. Paul grew up in England, as you'll notice once he starts talking from his wonderful accent. But then did a fairly traditional transition from England to Kansas, I assume. And did his master's in botany there before he went up to Washington State to do a PhD in botany. Did a short postdoc at UC Irvine, and then ended up quickly taking a faculty job at Utah State, where he's been ever since. In 1992 he started and got tenure in 98. Paul's been fairly well funded and published across his whole years, both in what he's going to talk about today, plant genomics, but also in pedagogy and education research. He has an NSF grant. I can actually spin this a little bit for Huntsville. He has a NATO grant. He has a Department of Defense, or has had Department of Defense grants. Where is he from? He has United States Army Corps of Engineers grants. So he has a lot of military-themed funding. But he also has U.S. Fish and Wildlife, NSF. And then lastly, one that stuck out to me was a 1.8 yen grant from the Japanese ministry. I'll let you guys put that into the conversion to see how much that is in American dollars. Paul has a number of papers, including a couple of ones that stuck out to me as for witty puns, and so hopefully maybe a lot of witty puns in this talk today. 2000, he wrote a review article about fluorescent proteins and probes called 1,000 Points of Light. For those of you young students here, you might have to Google that with Dana Carvey afterwards. And then he just had another review recently called Between Two Ferns. For you old people in the audience, you're going to have to Google that, so don't do it at work, I promise you. And you're going to talk a lot about ferns today and his research in that. So with no further ado, welcome to Huntsville Paul.
Thanks, Eric. Well, first, thanks to all of you for hosting me here. I'm having a wonderful visit, enjoying being in Alabama for the first time. What I want to do today is try and give a brief overview of my research program. So rather than going into lots of details, I want to try to summarize the organisms that I study, the kinds of scientific questions that interest me, the kinds of techniques that I use. And then I'll hopefully digress at some point to describe my approach to teaching, because like many graduate students in the past, I didn't learn how to be a teacher. I learned how to be a teacher by making lots of mistakes and eventually meeting people who knew how to teach and helping me learn how to teach. And then I'll finish up with some aspects of science that are especially, I feel, important, not just to me, but I think are important to science in general, and that I use as a focus for mentoring my students. So, not the only, but the main group of organisms I study are ferns. And someone asked me, when did I, and this was asked at the phone interview, when did I first become interested in ferns? And I think it's one of these things where I was never not interested. I just tend to accumulate interests and never lose interest. But since a child, I've always been fascinated by them, perhaps as a function of something ingrained in English culture. When we imagine ferns, we tend to have a picture like this in our heads. They're lush green understory plants, and that is actually true. Most ferns grow in both temperate and especially in tropical forests as a major component of the understory. That's not the only place you find them. You can find many species in extremely harsh, dry deserts, and you can find them floating in the water. This is a fern, Azolla, growing in a rice paddy, and it's been used deliberately in rice paddies for at least 1,500 years in China. And rice production today, most rice production involves the need for growing this fern because most soils and waters that are used for growing rice in rice production are low in nitrogen. And this fern, Ixola, has an endosymbiotic cyanobacterium that is used essentially as a biofertilizer. The fern is also being used for a lot of other functions, for generating fertilizer for other crops as well. It also conveniently has a relatively small genome, especially by fern standards. And so on my last sabbatical, which started at Duke University, I helped initiate the first fern genome project. We're getting very close to getting the first few publications together. We're tripping over assembly for some expected reasons, but a few unexpected ones, one of which is it's full of these cyanobacteria, but more endosymbionts than we had expected, which you'd think wouldn't be a problem because you can assemble those genomes separately, but there's been a lot of gene transfer and active gene transfer between host and endosymbiont, and it's actually tripping us up a little bit in the assembly, but in a very interesting way. I will be getting back to this one a little later, but as I said, it's not typical. Ferns have fairly large genomes. The average fern genome, from what we can gather, is probably about around 10 times the size of the human genome. There are many more chromosomes. Flowering plants, the average across flowering plants is about 16. Ferns typically have about 4 times as many. This particular fern, Addiscan fern, the genus Ophioglossum, I'm sure there are several species in Alabama. There's one in the Himalayas that has 1,400 chromosomes. There are several fern species with over 1,000 chromosomes. So chromosome numbers are very high, suggesting that they are polyploid. And based on the base chromosome number in this genus, this would be 84 ploid, which is quite high. So before diving into why ferns have high chromosome numbers and what that means for fern genomes, I'm going to digress a little just to remind, because it's been a while since any of us have taken intro biology, the different types of life cycles you get in plants and how ferns differ from seed plants. So this is a seed plant. It probably is for this. Looks like, no, actually it's not a seed plant. It looks like Selaginella, which is a lycopod. But heterospermy is common throughout the seed plants, the gymnosperms and the angiosperms, and that's simply where you have 2 different types of spores. You have the little microspores, and the spores in plants, of course, are the product of meiosis, so they're haploid. Microspores will grow up into these microgametophytes, and so in a seed plant, the microgametophyte is essentially the pollen, and it contains haploid nuclei. Meanwhile, the much larger megaspore grows into the megagametophyte, which in a flowering plant is microscopic, only about 8 nuclei. Fertilization occurs with one of the haploid nuclei from the microspore being taken up by the megagametophyte, reconstituting the diploid zygote, and that grows up into the embryo and the new sporophyte. So this is the alternation of generations you get in all plants. Most of the plants you see looking out here are heterosperous. They're doing this right now, especially in the spring. But most ferns are a little simpler in this respect. They produce one type of spore. They're homosperous. That single type of spore is produced again by meiosis. You look underneath the leaf of most ferns, you'll see these splotches that are the sori containing sporangia in which meiosis occurs. The spores grow up into one type of gametophyte, and that gametophyte, of course, is haploid, but these are much larger than those you see in seed plants. You can see them, you know where to look. In nature, they're typically about a millimeter across, but you can grow them in culture to be over a centimeter. And they can potentially produce both egg and sperm, which is a really useful tool in genetics because this thing's haploid, and if it can self-fertilize, if a haploid can self-fertilize and produce a sporophyte, it will be homozygous at every genetic locus. So it's a very useful tool for genetic studies. In nature, it turns out most ferns don't do that. It'll be a sperm from a different gametophyte fertilizing. Most ferns in nature are outcrossing, but in the lab you can get them to self-fertilize. That will reconstitute the diploidy again. Here's a photograph in the lab of a gametophyte. This gametophyte is probably about 4 millimeters across, and you can see fertilization has occurred, and now you can see the diploid sporophyte growing out of it with some vascular tissue. Now, most ferns are homosporous, and the one exception is this one little clade comes out near the middle that includes the heterosporous ferns, and those are all aquatic, and those include Azolla. And for some reason, the heterosporous ferns all have relatively small genomes, and all the homosporous ones tend to have very large genomes. They also have large genomes and large high chromosome numbers. Heterosporous ones tend to have low chromosome numbers and small genomes. The reason for that association between genome size, chromosome number, and the type of spore production is still a mystery. So, still one of my goals in research is to solve that association. Why do homosporous plants have more chromosomes? Over the last 30 years, a number of hypotheses have been put forward, some of them extremely satisfying, but they turned out to be wrong. So, as far as I'm concerned, we still don't have a satisfactory answer. So, great, we're sequencing a heterosporous fern, and it's really for very much the same reason that Arabidopsis became a model for plant genomics in general, a nice convenient sized genome. The other reason we're using Azolla is because it has some economic use. The Beijing Genome Institute are funding a lot of the sequencing because of their interest in rice production. But the problem is we're going to end up with a genome sequence for something that is not a typical fern. So in parallel with collaborators at the University of Florida, I'm working on sequencing genome of Ceratopterus, which is also being referred to as the Arabidopsis of ferns, but for other reasons, because it's a very important experimental organism for studies of development. It's used heavily in education. I talked about the life cycle of ferns. You can grow gametophytes of almost any fern species in the lab very, very easily. You can take a gametophyte, put it under the microscope, and you can watch fertilization. So for teaching aspects of plant life cycles, sex, fertilization in general, you can see sperm swimming up to the egg. You don't need any kind of special equipment. There's some really nice videos on YouTube taken by holding a smartphone camera over a cheap teaching microscope. So they're great tools. The sea fern Ceratopterus is homosporous, large genome, typical fern, and because of its use in research, we're working on trying to assemble this very complex genome with a relatively high chromosome number. But what use is a fern genome going to provide to the research community in general? Most of the genome projects are associated with organisms that have some kind of economic value, something that's on your dinner plate or is at least useful for some kind of human value. Well, the value in ferns is they are extremely useful contrasts for aspects of plant genomes in general and understanding the evolution and structure of plant genomes. They're highly polyploid, or they appear to be because they've got lots of chromosomes. Well, most plants, about 50% of flowering plants have been estimated to be polyploid. Even some animals are polyploid. Most crop species are polyploid, not every single one of them. And those mechanisms of doubling the genome, whole genome duplication or polyploidy, has been studied really well. What hasn't been examined in quite as much detail is what happens between these cycles of polyploidy. If plants over their evolutionary history keep doubling their chromosome numbers, well, they're going to get bigger and bigger and bigger, which means something has to happen between these cycles. And this is where studying ferns can be quite useful, because I'll be presenting some evidence in a moment that shows we think they contrast quite drastically to seed plants. Need to back up a little and talk about how the research has gone up till now, because most of the focus in general, not just in ferns but in plants, has been on the chloroplast genome. And the reason for this is the chloroplast genome is tiny, 150 kilobases on average. And so it's nice and compact, it's full of important photosynthetic genes, highly conserved in structure, and I've spent about the last 15 years working on looking at the structure and evolution of chloroplast genomes, mapping some of the inversions that have occurred throughout evolutionary history, and using the sequence of the different genes in this genome to reconstruct the phylogenetic history of ferns. And so we have a pretty robust framework now for fern evolution. Let's see if I've got a pointer. So there's a little group here, a little clade here that is representing the heterosporous ferns. You can see Azolla there. Everything else is a homosporous group. There, the tree fern clade there. And then this group up here, even though it looks fairly small in terms of the number of representatives in this tree, that actually represents 80% of the species of ferns. There are about 11,000 fern species. Most of them are up in this Polypodiales group that are radiated relatively rapidly in the Cretaceous. So it's a nice clean phylogeny. Everything looks really clean and simple. And that's because the chloroplast genome itself is relatively simple. When weird stuff happens, like hybridization, only one of the chloroplast genomes from one of the parents gets inherited by the hybrid. Whereas a nuclear genome is inherited by both parents, and so if you were to look at a nuclear genome phylogeny, it'll start to get a little bit messy. And it's only recently that we started to do this. So there's been this shift over many years ago now in flowering plants, and finally the fern biologists, we tend to be a little slow. We're catching up now and starting to look at nuclear genomes. And I started this a couple of years ago by just exploring the genome space. I wanted to try to document what was the just general content of a fern genome. So they're much bigger, they've got more chromosomes. How else do the fern genomes differ relative to genomes of other plants? So I looked at that phylogeny, picked some representatives from some of the major clades, one from the modern ferns, one of the tree ferns. I included, bottom left here, this extremely charismatic modern organism, the Ceratopterus. Fern biologists actually say it's the Arabidopsis of ferns because it's kind of so uninteresting in its morphology. But some of these other ones really are quite striking plants. But I chose them also because other researchers are doing some very interesting biology on these other species, and so the data hopefully could provide some extra benefit for them. Some of these had already had their genome size measured, others we need to do some flow cytometry to estimate genome size. That way when you get the data back you know how much coverage you've got. And many aspects of fern genomes were kind of not surprising, or were not drastically different from from other plants. The ribosomal RNA gene content was fairly typical. We were finding lots of extra copies of genes, which we kind of expected, but because it was low coverage, we didn't really know. We were getting at about 1 to 2% coverage. So sorry, 1 to 2x coverage. And between about 5 to 10% of the genome was getting covered. We also found a lot of genes that we did not detect in sea plants, but that also wasn't too surprising. We found a lot of animal genes associated with flagellum assembly, but that's not too surprising because sea plants, or most sea plants, the cycads have motile sperm, but all the flowering plants do not have motile sperm. Ferns do, and so it wasn't a surprise to see all these genes that are known to be associated with assembling the flagella apparatus. The main area where we found ferns to be a little different from other plants was in their repetitive structure. So we used several tools to explore the repetitive structure of the fern genomes, and we compared this with analogous analyses in seed plants. This has actually been repeated by other researchers who've now added several different additional ferns and tending to see the same patterns. So their genomes are larger, and one of the reasons they're larger is there's more repetitive structure in fern genomes. That's going to be a problem for assembly of these large genomes. But there were 3 components that consistently are higher in ferns relative to seed plants. One of the lines, the long interspersed nuclear elements, and these are mostly non-functional retrotransposon fragments. About 20% of our own genomes is made up of long interspersed nuclear elements. And they're typically higher in content in animals than in plants. So ferns in this respect are a little bit more like animals. They also have higher levels of DNA transposons and simple repeats. So these are short repeat structures like microsatellites. So more repetitive structure content in general, but particularly these 3 components are showing up to be much higher in ferns than in seed plants. So we were seeing these extra copies of the genes, but it still wasn't getting at that question: is there a signature of polyploidy in the fern genome, and can that be used to actually study the process? And so what we wanted to do was to start looking at other ways of studying polyploidy and getting a better handle on the actual genes themselves. When you do a shotgun sampling of the genome, you're capturing everything, which is useful if you want to see the average genome content in repetitive DNA. But if you want to focus on the actual functional genes, it's a little easier if you look at the expressed genes, the genes that get transcribed. And so in collaboration with Mike Barker at the University of Arizona and Josh Durr, former graduate student of mine, we've been looking at tens of thousands of expressed genes in ferns using transcriptome data, and then we start screening the data for gene duplications. So when a gene gets duplicated, that can happen in a lot of different ways. It's very common for just a gene to get a local tandem duplication. And when that initially happens, the copy is going to be identical. If those copies remain and diverge over time, eventually you're going to start to see differences emerging. And the longer those 2 sequences diverge, the more different they're going to be. And so you can use sequence divergence as a proxy for time. So if a few of these tandem duplications occur, you'll get a little bit of a signal. But if every gene in the genome gets duplicated, what you'll see is kind of a peak in the distribution because lots of duplications occurred at that level of sequence divergence. This has been done to plot, it was actually initially used to plot the amount of divergence, the amount of polyploidy, ancient polyploidy, even in something like Arabidopsis, which has a tiny little genome, but still has a signature polyploidy. And sure enough, in the ferns, we were able to see these, so this is essentially the amount of, it's actually synonymous substitutions in terms of sequence divergence. But you'll see these peaks, the peaks with, in the distribution where you see a high frequency of similar genes indicates a relatively recent polyploidy event. If you take a species that has a double the chromosome count of most of its relatives, it will have one of these extra peaks on the left. But then you start to pick up these ancient, older polyploidy, whole genome duplication events. We mapped this onto our fern phylogeny framework, and sure enough, you could see where on the phylogeny ancient duplications have occurred. This has all been done extensively in seed plants, gymnosperms and angiosperms. So we were able to compare the amount of polyploidy going on, and sure enough, not surprisingly, these ferns with these very high chromosome numbers are polyploid. But in general, if you look at the number of polyploidy events that have occurred over time, they don't seem to be any more polyploid than seed plants. You see about the same number of ancient genome duplication events as you do in both gymnosperms and angiosperms. And yet, angiosperms don't keep accumulating chromosomes. Ferns do. So what we're hypothesizing is that it's this part of the cycle between polyploidy events that's different in ferns. Angiosperms have all kinds of mechanisms probably associated with meiosis, but we don't know for sure, where you can get loss of extra duplicated material, and that can happen very quickly. There's a few research labs that are monitoring the loss of genetic material in polyploids over just a few generations, and it happens very quickly. And what I want to do over the next few years is do this in artificial polyploids in ferns and kind of measure over time the amount of genetic loss and see if I can get a handle on what those mechanisms are, the mechanisms that the angiosperms have that ferns do not. So it looks like you get slower loss of this genetic material between the cycles of polyploidy. One of the results of this is that it explains why in most groups of organisms there's no good relationship between chromosome number and genome size. It's really poorly associated in animals, and in most groups of plants there's no association, except ferns. The correlation between genome size and chromosome number is actually quite high, which is consistent with the chromosome number being a function of the number of polyploidy events rather than what happens between them. So ferns are a little weird in terms of their genome dynamics, which makes them a good kind of contrast for comparative studies of genome evolution. They're also a little weird in some other aspects of their life history. So a colleague of mine, Carl Rothfels at UC Berkeley, led a study a few years ago looking at a NAFL hybrid, a fern hybrid found in the Pyrenees. And they found very strong evidence that it's a hybrid between two different genera that have diverged by 60 million years. Okay, so maybe this is a one-off thing. So we started looking into the literature for well-documented hybrids and plotting that by then taking the parents of the hybrid and mapping that onto a time calibrated phylogeny, you can estimate the age of the node where the two parents diverged. And there are some hybrids between genera beyond 60 million years, and these are just the ones with really good evidence in the literature. Obviously, the closer the parents, the more likely they are to hybridize. Now, in an angiosperm, if you see a hybrid between two species that have diverged by over 1 million years, it's relatively unusual. So, the weirdness in this respect is probably a function of what goes on with fern sex. This is a two-fern archegonia. So, the egg cell you can actually see right here on the left. And you've got the archegonium neck, and there's a little opening there. And the archegonium releases a chemical attractant, and the sperm will swim over, swim down the neck, and fertilize the egg. Very simple. There are no, in angiosperms, you have all these mechanisms of controlling how the sperm cell can get to the egg. You've got pollinators, you've got complex events controlling the timing of sexual maturity. None of that exists in ferns. You've got sperm swimming over to the egg. That chemical attractant of most species will attract sperm of ferns. And there's very little differences between the species. The chemical that attracts the sperm will work on a lot of different species. There are other pheromones being released